Just to introduce myself, as part of my role, I work with a life science data infrastructure called Elixir UK, with a mission to support the management and analysis of life science data. So the UK node feeds into a larger pan-European network of a number of countries. But as part of the work over the past years, we've been looking at supporting researchers in doing better data stewardship and managing their data appropriately. So for this talk, I wanted to signpost resources useful not just for life sciences, but also across disciplines, but also to say a little bit about how we've built training and, for want of a better word, products that help people in the life sciences manage their data better. So historically, there's been a large focus around helping the researcher manage data, but really eyes are now on how data stewards and RTPs can be better supported. So the term FAIR is now everywhere. So the Medical Research Council, the MRC have a new data sharing policy as of last year, and there is no mention of open data anymore, but instead mention of FAIR data. So another example of a large funder, Cancer Research UK, ask their applicants to define how they will make their data fair. UKRI itself released a piece last September on ensuring open research, and mention FAIR explicitly, and also UK government. So I'm just giving an example here of a document which is their national AI strategy, which not only mentions FAIR, but mandates. So we're starting to see this coupling of AI and also FAIR. So I don't want to spend too long on defining FAIR, particularly for an audience who will know what it is, but FAIR is an acronym, stands for findable, accessible, interoperable, and reusable. A lot of people get the R wrong. I've seen this a few times. The mistake people sometimes make is they think that the R is reproducible. In fact, the R of FAIR is reusable. But FAIR can apply to a lot more than data, but quite commonly is used in the context of data. So we are now seeing things like FAIR workflow, for example, and also something that Elixir does a lot, which is how to verify training material. So there's quite a nice white paper from Elixir around how to verify a piece of training, so how you can actually make the training itself findable and accessible and so forth. But FAIR defines 15 principles, and where you address these, you are effectively verifying your data. So, for example, the first is that your data is given a unique and persistent identifier, which usually means, well, certainly for my field, you can supply a URL or DOI to your data, and that this URL exists forever. So where life science really has embraced FAIR is that it permits closed and restricted access to data, which is essential in instances where we work with sensitive personal data that cannot be shared. So this isn't unique to the life sciences, but is a large consideration for us. FAIR also permits us to delete data. So in this case, the 8th principle comes into play, where you can remove the data from the link, but the link must remain active, as well as the metadata describing the data that has been removed. So Zenodo works like this. You can delete the data, but you cannot delete the metadata from the DIO, which should now contain a reason for deleting the data. So we see this in the biological databases where certain genes or proteins become deprecated. The link will be persistent, but there will be a note there that a certain record has merged with another one. So the table here comes from the publication, the FAIR Principles, which is quite often not cited when talking about FAIR, but instead people quite often get the top Google hit, which is the Go FAIR pages, which are really good. So the reason I wanted to show this is because it kind of starts to put FAIR into context. So for each of the FAIR principles, it gives examples of each of these. And this is just a page which is showing examples of what a persistent identifier is. So when I last looked at this was very much life science focused. But if I wanted to show a novice really of FAIR what FAIR actually is and what it actually means, then this would be one of the first resources I think I would show. So from here on, I kind of want to talk about the work I've been involved in within Elixir UK. I know we have Phil Reed speaking about Elixir output today and tomorrow, so we've made sure we don't duplicate information here. So I'll leave a discussion of what Elixir is to Phil. But instead, I'll talk about the FAIR fellowship we ran community and its outputs. This was our first grant, really, as a node entity, where I led with another PI from Bradford, though this had a wider delivery team. So there were a team working on this, leading the interlocking parts. So UKRI released a training call for applications to upskill researchers in the life sciences in a thing called bioinformatics, which is kind of a catch-all term for compute, data stewardship, RSE within biology, but also looking to upskill researchers in AI. And we as a node received an award where we partnered with the Software Sustainability Institute to build a fellowship of FAIR data ambassadors to themselves train within their own universities and communities. So SSI support a fellowship running for many years of research software engineers, and we partnered to do the same around data stewardship in the life sciences. So fellows were supported to become local data steward champions within their universities and communities, and in this case focused on training activities. So the idea is that since training is delivered locally, we expect content and know-how to become embedded locally, and through local embedding, training then becomes sustained and becomes part of the institution's staff curriculum. And really, this is the holy grail, or one of the holy grails of training, is that training that can be found and that is then embedded within the university curriculum. For example, the courses around research data management that they run for their own people. And this is in fact what we are seeing now. So local data management teams are signposting to some of the courses that I'll show you now that we've written in the life sciences and healthcare, as well as other resources such as RDM Kit that I'll show you as well. So these data stewards are in a network. They will amplify the activities going on without having to coordinate all the comms really from a single place. So an example of this is the University of York, who gave a piece of training around the stewardship of cancer data. And they had 60 people sign up, half of which in fact came from Wales. I fed the information into a Welsh biology network we have. So this kind of idea of, bums on seats, as my father would say, hasn't really been a problem because of this active grassroots network. So fellowships aren't unique to Elixir UK. There's an Alan Turing-led award, People in Data. They're looking to disseminate activities through a Champions programme. Also Eurobioimaging, which concerns itself with image data in the life sciences and is across Europe, and now running a fellowship or ambassadors programme to do the same sort of work. So we have 24 fellows across the UK through 2 rounds of recruitment. The fellowship supports a mentoring programme as well as Train the Trainer. So fellows support locally within their fields of interest. They swap material with others and get new material into their organisations. And this is kind of part of a professionalisation process where the data stewards are recognised by the universities. They become visible within the university through doing this work, and become kind of a go-to person when data stewardship and management is being discussed. So our partners in the Netherlands are looking to do something similar within their country around building and supporting data stewardship in the life sciences. So the fellowship was interested in building short courses, so a short carpenter's course to position FAIR. When we looked, And Elixir does this a lot. It doesn't really do what's out there already. It will signpost to what's there, but where there are kind of holes, they will start to resource and build and fill these gaps. But when we looked for training around FAIR and life science data, there were really good resources, but the training in terms of things with learning objectives and formative assessment and constructive alignments were pretty thin on the ground. And what we found was, what we did find around FAIR courses is they were pretty high level. They were talking about why FAIR is useful and why we should actually share data, rather than going into the practitioner knowledge of how FAIR actually works and how to verify a piece of data. So what we did here is we took a steer from two of the FAIR authors, so two of the people on the authorship list of the original publication, who talked about moving the principles around under 4 headings. So here, just on the right, we've got the metadata, data registration access, and persistent identifiers. And what we did is we moved these principles under these headings. And I've just shown how, metadata, for example, appears throughout the F, the A, the I, and the R. So here we can have a chapter to each of these and pull in the relevant principles under a single heading. So this is what we've done. So a course here addressing the headings which bring in the 15 FAIR principles. So fellows have written this, fellows have taught this and signposted within the university intranet pages. Certainly in Cardiff University, where I am, we point to this, and also another carpenter's course written in Edinburgh, which is excellent, and again looks at FAIR from a slightly different angle, showing what can be done to better manage life science data. So it kind of goes into, how you name a file, how you aggregate and collect data and organize that data, best methods for collecting reproducible provenance as well in terms of the metadata. So to complement this, we wrote another course addressing a specific data type called RNA-seq. So we didn't use carpenters in this case, we used the Galaxy training network, where again we have strong links into the infrastructure. So RNA-seq is one of the life science omic data sets where effectively you assay for the abundance of RNA in cells in the body. What you do is you get a landscape of gene expression. So effectively you can identify genes that are turned on or off in a state, for example, a disease state relative to normal tissue. So we took this data set and we kind of followed these headings. So we showed metadata, particularly the metadata community standards around annotating RNA-seq data sets. And then we showed the public repositories, which then cater for the registration, the access, and also the persistent identifier. So we scaffolded against the previous course that I've just shown. I think this is one of the useful areas for the future, which is data stewardship around specific data types. I'm working currently with an EOS task force where we are mapping the data types across domains. It's pretty high level. I think there's a need for a bit more granularity under these data types. But it's just so that we know the landscape. Also, as part of the ambassadorship, so the fellowship, we set up meetings with the data leads at fellows universities to really discuss, what was needed. So what is being done well by universities, and we really found that generalist support was really well performed. People were running courses and these were very well attended. But in terms of real sort of blind spots or things that were missing, people were talking about access to more material around specific data types. So the beauty of the fellowship and organisations like Elixir UK is that you can invite people into hackathons and start to build pieces of training, and practitioners have written this work. So another example of where co-creation is working in the larger Elixir space, and here we go into Europe, is the RDM kit. So again, we are finding universities signposting to this. This has been signposted to also by the Horizons programme as a recommended data resource in the life sciences. So this is a toolkit. research data management toolkit for the life sciences, but really has a lot of information that is applicable across domains. So it's written by data stewards, it's written by practitioners, around 100 people so far contributing to this, and it's built very much around the style of the Turing way, if you've seen that. So there are pages on the data lifecycle and specific pages addressing different data types, but also communities, with signposting to useful resources, community standards, and also training. And this is what I like. It's really the aggregation and signposting of everything that is out there, and there is a lot out there. So pointing to things and not redoing them. So Elixir have a training registry called TES, where we register a lot of the training that is out there, and we've linked these two resources together so that they link to each other. I wanted to show you too some of the short videos we wrote as part of the work. So one of the fellows at Queen Mary works as a data steward within a large consortium, and detailed how she uses the data catalogue to keep track of the metadata and the ownership within a consortium producing many data types. So I use these all the time. So this is kind of an example of where I've used some of the outputs and started to customize within my own university, where we now have a local data steward club, and people are creating data catalogues for their own teams so that PIs know where their data is and know where the data has actually come from. This is kind of an example of a real shift in culture. People sort of talk about, cultural change around data. And this is one routine that I've got with some of the PIs I certainly work with. And so when they need to find their data and the metadata, they are not going into their email or into the data repository and searching for stuff. They go to this data catalog, which is effectively a spreadsheet, and you find everything you need there. Where the data came from, how it was produced, provenance, who archived it, dates, and so forth, and the locations of where the data actually is. So finally, I just wanted to signpost to recent-ish fair developments in the life sciences. BioFair was funded last year and has had Elixir UK involvement, though it's a separate project. It's a UKRI, DRI led by BBSRC. So the effort is to start linking the UK data management landscape in the life sciences with commons and services for software, workflow data and communities. So this is very much echoes what is happening in Australia with data and life science data, where there's a linking of resources. So I won't say too much about this because I'm not too sure on the granularity which is actually being decided currently. But the effort here is to use what is there already, so that kind of getting to a point that anyone in the UK can find data in other locations, not just local and in the public databases, workflow and people within a distributed ecosystem. This came out last week, so this is in Nature. It's cross-discipline, describes FAIR in the context of computational workflow. So I've only skim read this, so I need to give it another read because it has some really nice examples of how to verify workflow and how fair workflow work with fair data. So one of the cases includes a thing called the RO crate, which is one of the digital objects which is being used to share data and metadata between repositories, and also sensitive data between repositories. So finally, we have an RDM club in Elixir. We welcome anyone who would like to join. So we have monthly calls. Yesterday we had UCL present on their data stewardship group and how their journey really from inception to having a group now of around 15 individuals. We had around 60 people on the call, so this was a popular one. The mailing list is low traffic. We won't fill up your inbox, but it lets people know what's coming up. High traffic is on our Slack channel, and people can join this too. Thank you very much.